Hi, my name is Ashwin Silva, and I'm a final year undergraduate at the Department of Electronics and Telecommunication Engineering at University of Moratuwa, Sri Lanka. And I'm the lead author of the paper titled Real-Time Hand Gesture Recognition Using Temporal Muscle Activation Maps of Multi-Channel sEMG Signals. We will start off with our introduction that includes previous work, shortcomings of the previous work, and our contributions. And then we would have a look at the intuition behind our method. And then we would explain our method in detail, and it will be followed by our results and conclusion. The sEMG signals from the forearm are widely used in gesture-controlled applications and prosthesis control systems. Most of these methods rely on binning the sEMG signals and computing a set of features such as mean absolute value, waveform length for each of these bins, and labeling these bins using a trained classifier such as support vector machine, LDA, and hidden marker models. Then we can look at the work by Kithmin et al., who uses continuous binning approach as well. They classified each of their bins using an LDA. They also used optimal positions on the forearm to place electrodes so that they can tap into the muscles to access the good signals that are pertaining to a certain hand gesture. They treated the channels as individual and uncorrelated entities, and they did not consider the mutual and correlated aspect of the channels. In this figure, you can see the continuous binning approach that they had used. Furui et al. Also worked in hand gesture recognition, and their approach was based on muscle synergy. They also used a continuous binning method, and the classification was performed on each bin using a log linearized Gaussian mixture network. Like Kithmin et al., they also used optimal positions on the forearm to place their electrodes. And quite differently from the Kithmin et al.'s work, they considered the channel correlations and the mutual aspects of it as well. In this figure, you can see the approach that they had taken. With that, we can take a look at the shortcomings of these methods. The continuous binning causes real-time gesture predictions to be computationally expensive. This is because the algorithm has to predict a label for each of these signal bins, despite the fact that there might not be a gesture onset there at all. And this can take a toll on the computational efficiency of the algorithm, and it is highly important to maintain a speed if you are dealing with the real-time application. And second shortcoming is that most of the previous work on real-time hand gesture recognition have focused on extracting the features by considering sEMG recordings from the electrodes as individual and uncorrelated entities. And we hope that if you look into the mutual aspects and the correlated aspects of these channels, you might be able to glean some more information about the hand gestures. And then with that, we can introduce our contributions in this paper. The key idea that we are proposing in this work is the temporal muscle activation maps that can represent both the individual and mutual activation patterns of the forearm muscles. And also, based on these TMA maps, we also introduce a novel gesture onset detection method and a novel computationally efficient real-time gesture classification algorithm based on TMA maps. With that, we can take a look at the intuition that we had behind our method. And in this figure, you can see an sEMG recording obtained from the myo armband positioned around a circumference of the forearm. And there are two onsets present in this figure. Onset A corresponds to the flexion of the middle finger, while the onset B corresponds to the extension of the middle finger. And if you monitor closely, you can see that the signals exhibit a correlated change across the channels each time a hand gesture onset is occurring. To visualize these changes in the channels, we extract the envelope of the sEMG signal. An envelope is a measure of the amplitude modulation of a signal under consideration. And the signal amplitude of a sEMG signal represents the activity or the tension of the muscle mass. In the blue color lines in the figure, they represent the envelopes of the signal that you saw in the earlier slide. Therefore, the correlated changes in the envelopes of the sEMG signals across a given set of channels represents the pattern of activated muscles related to a hand gesture. This is an important observation in our work. So these coordinate changes were described in a temporal muscle activation maps, which is the key contribution in our paper. We sought out for a method to describe or represent these coordinate changes in a mathematical manner, and we came up with this novel idea of TMA maps. And they are able to quantify the temporal changes in individual and mutual muscle activations within a given time window. In this box, you can see the mathematical definition of TMA maps. First, you take a time window across all channels, which has a length of sample size m. And then for each time points in this window, we compute a vector called A, and then which includes the first order terms and the second order terms. The first order terms represents the individual nature or the characteristics of the signals and channels. The second order terms represents the mutual connections and relationships between the channels in the sEMG signals. And then after we compute these vectors for all the time points in that window, we order or we concatenate these vectors according to their temporal order to arrive at our matrix, which we call the temporal muscle activation maps. This process is well represented in this illustration. We have L number of signals, and then we take a time window with a length of sample size m and at a position of n naught. And then we compute this an vector, which includes the first order terms and the second order terms for all the time points in that particular window. And then we organize them according to their temporal order to come up with the temporal muscle activation map at point n naught. And now we have seen that the TMA maps are capable of representing the pattern of muscle activations, both individual and mutual, in a compact manner. Then, if you consider a time series of TMA maps, we can say that it is an estimate of the muscle activations over time. And this is important when it comes to onset detection. We know that whenever a gesture onset is occurred, we can observe a correlated change in the sEMG signals across the channels. Therefore, a significant change in the time series of TMA maps can indicate the occurrence of a gesture. To quantify these changes in the TMA map time series, we define a signal called difference signal, denoted by dn. This dn is defined as the difference between two adjacent TMA maps. And to convert this matrix change into a scalar value, we use the Frobenius norm. Here you can see a picture of the difference signal plotted against the corresponding correlated change or the gesture onset. And when we have a correlated change like this or a gesture onset, you can see that the dn has a higher response. And this point of higher response can be detected using a predefined threshold. And this point of higher response is considered as the gesture onset. And after an onset was detected, the onset detection procedure was paused for a predefined duration, which we call the refractory period, or R. And this is imposed in order to minimize the false positive onsets. And then another important part of our algorithm is the classification of these TMA maps into different gestures. TMA maps at the time of the gesture onsets can be used as the training data for a classification algorithm. And since there is an image-like nature to the TMA maps that can represent the temporal variation of both individual and mutual muscle activation patterns, we can use a powerful computation tool called Convolution Neural Network, or CNN, to classify our TMA maps according to its gestures. And here in this picture, you feed a TMA map to a trained CNN to extract its features, and then using a fully connected neural network, we can predict the hand gesture. This algorithm sums up what we are doing in our method. The important part of this algorithm is this if clause. It has two conditions. In the first condition, it says that it must detect an onset because the dn should be higher than the specified threshold. And then at the same time, you have to meet the condition where the elapsed time since last prediction should be higher than the refractory period R. And then only when both these conditions are met, we send the TMA map at that particular time point of gesture onset to our CNN, which is trained, and then this trained CNN can predict the gesture label of that particular TMA map. And this method, as you can see, does not make a prediction for all the time bins that we compute. Instead, it only takes in the time points where there's a gesture on set and then only classifies it according to the label. And this increases the speed drastically, and also it can increase the accuracy of our algorithm as well. And with that, to explain about the experiments and analysis, I would hand over the presentation to my colleague, Malsha.
Hi, I'm Malsha Perera. I'm a final year undergraduate of the same department and the same university as my colleague Ashwin. All the experiments were performed on 8 healthy subjects, and the sEMG data was obtained using the commercially available myo-arm band. And the myo-arm band was placed along the circumference of the forearm as shown in this figure, and the arm was placed on the table when collecting all the data. And in this study, we considered 5 gesture types: pointer, middle flexion, ring flexion, V flexion, and the hand closure. And we asked each subject to perform 20 repetitions of each gesture, and the gesture was held for a period of 5 seconds, followed by a resting period of another 5 seconds. And we trained individualized convolutional neural networks for each subject. And these were evaluated in a real-time study of random sequence of 150 gestures. And we ensured that each gesture type would have an equal number of repetitions. If we take a look at our results, we were able to get an average computation time of a gesture prediction around 5.5 milliseconds on a 2-core, 2.4 GHz Intel Core i7 CPU. If you take a look at the table here, we compare our results with the state-of-the-art methods available in the literature. Our method was able to recognize middle flexion and ring flexion with higher accuracies than the state-of-the-art methods. Compared to the study conducted by Kappel, the classification accuracy of our method was higher for all the gestures. When comparing with the study conducted by Furi, it is important to note that the myo band has this inherent limitation that all the electrodes should be placed on a common circumference. This limits the access to some of the muscles that are directly linked with certain finger motion. So even with this limitation, we were able to achieve comparable results with the study conducted by Furi. And they used the optimal electrode positioning, whereas we placed the electrodes along a circumference on the forearm. And in the chart at the bottom of the slide, you can see the classification accuracies across all the subjects. And you can see that all the subjects showed classification accuracy about 90%. And as the conclusion of our study, we would like to say that the TMA maps were utilized to detect gesture onsets and recognize 5 selected hand gestures in real time with an accuracy that was comparable to the state-of-the-art methods. Looking forward, it might be possible to increase the classification accuracy even further with more optimally placed electrodes. And now I would like to hand over back to my colleague Ashwin.
And with that, we can conclude our presentation on our paper. At the end, we would like to acknowledge the ICASSP 2020 Organizing Committee for moving the conference to a virtual platform due to the outbreak of COVID-19 pandemic. And also, we would like to extend our sincere gratitude to the Bionics Laboratory at Department of Mechanical Engineering, University of Mattua, Sri Lanka. Thank you for listening to our presentation and have a nice day.